In the previous lesson, I mentioned that evolution is a theory. It's not an opinion or a hunch or a belief. When we say that evolution is a scientific theory, that means that there is a large, extensive body of evidence supporting the concept of evolution. And so we're going to examine some of these various lines of evidence that have emerged within the scientific study of evolution. And most of what I'm going to cover is from pages 387 to 393 in the Campbell Biology in Focus textbook. So here are the various lines of evidence that I'll mention. Some of them I'll go into more depth on than others, but they're all important pieces of the puzzle that support this idea of descent with modification and also the idea of common ancestry of all life on Earth. And when you think about the evidence for evolution, I guess the most direct evidence that we have, without question, would be the fossil record, because the fossil record gives us snapshots of what life has been like many, many, many years ago, in some cases many millions or hundreds of millions of years ago. And it shows us very clearly that life on Earth today is different from life on Earth long ago. And in some cases, the fossil record actually can help us understand how modern species may have evolved from previous ancestral species. I'd like to begin talking about the fossil record by showing you this photograph of one of my very favorite fossils. This species called Archaeopteryx lived about 150 million years ago, and this particular fossil was first discovered, the first specimen of this species was discovered in 1861. And when you look at it, it looks like a pretty typical theropod dinosaur fossil. The theropod dinosaurs were the two-legged meat-eating dinosaurs. The most famous example would be velociraptors, where anybody who has seen Jurassic Park movies knows about velociraptors. And this particular member of the velociraptor family was a fairly small meat-eating dinosaur, probably not much larger than a wild turkey that you might see today. But when you look, you see typical theropod dinosaur features. You see a long reptilian tail, very large claws on the feet, as well as claws on the forelimbs, very sharp teeth within the skull. So it's a pretty typical meat-eating dinosaur, right? Except what's unusual about this theropod dinosaur is very clear in the fossil are the imprints of feathers that were on these forelimbs. So essentially, they were wings. Now, were they flight-capable wings? Scientists still debate a little bit about that. Perhaps this animal glided from tree limb to tree limb and maybe didn't truly fly in the traditional bird sense, but not only feathers on the forelimbs, but very clear imprints of feathers on the tail. So this is a great example of what scientists refer to as a transitional form. Archaeopteryx, this is kind of a modern artist rendering of what this animal may have looked like, clearly had features that match up with ancestral dinosaur species. I mentioned the sharp teeth, the long tail, the claws on all the limbs, but it also very clearly has features of modern birds. Feathers on the wings, feathers on the tail, which this particular artist rendering did not include, but many Archaeopteryx fossils very clearly show feathers on the tail. And so it's a transitional form. It's a species that gives us a little picture of what that transition from two-legged meat-eating dinosaur to modern bird may have looked like. So a transitional form is any fossil that shows features of both an ancestral species and a more modern species. And Archaeopteryx is a very good example of a transitional form. But the fossil record in general is very valuable to scientists seeking to understand how evolution has worked in the past. When we look at modern whale species that are on the earth today, none of them have hind limbs, and their forelimbs, their front appendages, are, of course, fins so that they can easily swim through the water. But when we look at species of whales that lived long ago that are now extinct, we see that many of them had forelimbs, both, uh, no pun intended, they had four limbs, forelimbs and hind limbs. Um, and if you go far enough back in the fossil strata, you'll find animals that were clearly whale ancestors that seem to have spent maybe only part of their time in the water, but still had limbs that were functional on land. And in fact, this species, the mesonychid, which went extinct about 36 million years ago, clearly was a four-legged land-walking mammal. But it has features in its skull. There are little bones in the mesonychid skull that are only found in one modern group of animals, and that's modern whales. So we think even this land mammal, this mesonychid, which never probably spent any time in the water, was a ancestor of modern whales. And so for a long time, whales were a mystery because they live in water like fish, but they're clearly not fish. They have lungs rather than gills. They're warm-blooded, whereas fish, as you know, are cold-blooded. And so it was like, how did mammals end up in the ocean? And how did whales end up looking so very different from most other mammals? And we now have this clear idea of how whales very likely, I would say almost certainly, descended from four-legged land-walking mammal ancestors. The fossil record provides us with that information. Speaking of whales and their ancestry, a lot of people don't realize this, but even a modern whale without any hind limbs at all still retains internal bones that are left over from the leg bones of its ancestors. So, for example, in a modern whale, you will still find pelvic bones, a femur bone, and even small lower leg bones. These are called vestigial bones because they serve no function. They're just relics of the long ago ancestry of that modern whale. Another line of evidence that provides very strong evidence for evolution besides the fossil record is what we refer to as homology. And I'll explain that term a little bit more in a moment, but I want you to look at this image. We're looking at the forelimbs, the front limb, from four different species of vertebrate. These are all mammals. But in fact, all tetrapod four-limbed vertebrates have these same bones. If you look at the human arm, we have a single bone in the upper arm called the humerus. We have two parallel bones in the lower arm, the radius and ulna. bones in the wrist, which are called carpals, the bones of the hand, which are called metacarpals, and the bones of the fingers, which are called phalanges. But if you look at the front limb of any tetrapod vertebrate, you will find those same bones: humerus, radius, ulna, carpals, metacarpals, and phalanges. You see it in the flipper, or pectoral fin is the proper term, of a whale. You see the same bones in a bat's wing. We call this phenomenon homology. As you know, homo means sameness, or something's the same. So homology is whenever we see some feature that is the same amongst many different species. And presumably it's because all of those species share a common ancestor. There are many different kinds of homology that we'll talk about. There's molecular homology, there's embryological homology, but here we're looking specifically at structural homology. It comes from a discipline in biology called comparative anatomy, where you're looking for similarities in the anatomy of different species. And when we find those similarities, we refer to that as homology. Now, the reason all tetrapod vertebrates have the same internal bone structure, the same bones modified for very different functions, is because if you go back in the fossil record, say 500 million years, the only vertebrates on Earth at that time were fish. There were no amphibians, reptiles, birds, dinosaurs, or mammals. But about 360 million years ago, from fish ancestors evolved the first land vertebrates, which were amphibians. And those very earliest amphibians, those first vertebrates with limbs rather than fins, had a humerus, a radius, ulna, carpals, metacarpals, and phalanges. And those amphibians passed that bone structure down to their descendants, the reptiles. And the reptiles passed that same bone structure to their descendants, which were dinosaurs and birds. And eventually, reptile ancestors passed this same bone structure down to their mammal descendants, of which we are one, because as you know, we are mammals. And so the reason all tetrapod vertebrates have essentially the same bones is because they inherited that common bone structure from a common shared ancestor. So that similarity in bone structure is what we might refer to as homologous structures. Structures that are shared by two or more different species due to common ancestry. And whenever we see homology like this, it provides evidence of what we call divergent evolution. So notice we have, and this is sort of a hypothetical example, but we have 4 modern species. I'm calling them species A, B, C, and D. But if you trace their ancestry back, you'll see that they all share this ancestor in common. And so if they all branched from that common ancestor, even though these are different species, and they might look different, they might behave different, they might live in different habitats, we would expect all of them to share some things in common. Something should be similar about their bone structure. Something should be similar about their DNA, their protein structure, the way their embryos look. And if we have 2 species that share a very recent common ancestor, we would expect the level of homology to even be greater. In other words, the bone structure, the DNA of species C and D should be even more similar than, say, species D compared to species A. because A and D shared a common ancestor long, long, long ago, but C and D share a much more recent common ancestor. So homology is evidence that divergent evolution has occurred. With homologous structures, we see the same basic anatomy, but it may be highly modified to perform very different functions. So the bat uses these bones for flight. The whale uses the same bones for swimming. Four-legged mammals use those same bones for running and walking, and we, of course, use those same bones for playing the violin or writing or all the wonderful things that we can do with our limbs. Earlier, I made reference to these little bones that seem to serve no purpose in modern whales: a pelvis, femur, lower leg bones. Even though modern whales do not have functional hind limbs. So what this seems to suggest is that the whale has descended from an ancestor that did in fact have functional hind limbs. And so these so-called vestigial bones are just evolutionary leftovers. And they give us kind of a sneak peek into the evolutionary past of a modern species. So vestigial structures are yet another piece of evidence that supports evolution. Vestigial structures are any body structures that serve no function in a modern species, but must have been functional in an ancestor. So I mentioned the pelvis and hind leg bones in whales. We see the same thing in many different species of snakes. Now, as you know, snakes don't have functional limbs, but their skeleton still retains non-functional leg bones. Not all snake species have these, but many of them do. The human coccyx bone, which you know as your tailbone, is really a fusion of three small bones that have fused together right at the base of your spine, what's called your sacrum. And it serves no function in your body. But if you were a dog or a cat or a squirrel or a monkey, those would be the first three bones of your tail. They're what we call caudal vertebrae. And since we don't have tails, the bones have just fused together, and they're still there, but they don't serve a purpose. The human appendix is a tiny sac that's attached to the very first part of the ascending colon. So it's part of the digestive system, but serves no function in humans. Presumably our ancestors used the appendix for maybe to help digest uncooked foods, foods that were hard to digest, like fibrous plant material or uncooked meat and things like that, which we don't eat much of today. But we don't know exactly what its original function was, but it's there for a reason, and it's there because our ancestors had a use for it. Likewise, vampire bats, which are kind of a weird species of bat that lives, I know there's some in South America, maybe in Africa as well. It's a species of bat that feeds exclusively on the blood of animals, and they tend to find sleeping livestock, large animals asleep at night, and they'll swoop down and they'll make a tiny slit in the skin of the animal, and they lick the blood as it oozes out. Very disgusting. They feed exclusively on blood, so they don't need molars. They don't need to chew anything, and yet they have molars. Why do they have molars? Because bats are directly descended from rodents, things like mice and rats, and those animals use their molars. And so in vampire bats, they're completely vestigial, just leftover remnants of their evolutionary ancestry. Look at this picture. These are embryos, early stage embryos of 6 different species of vertebrate. Now look at the embryos. They all have gills, what we call gill slits, and they all have a long tail. Can you tell which one is the salamander? Which one is the tortoise? And get this, one of these embryos is a human embryo. One of these is a picture of what you looked like in the early stages of your development inside your mother's womb. Yes, you went through a stage early in your development. Before you had arms and legs, you had gill slits and you had a tail. So you guessed it. This is the human embryo. No, it's not. See, I fooled myself. The human embryo is this one. There we go. Now we can tell what's what. But my point really is that they all look the same. You know, as different as fish and salamanders and tortoises and chickens and rabbits and humans are very different as adults, in the early stages of embryological development, they look almost identical. Why do embryos look so similar in the early stages of development? Could it be because all of these vertebrates share a common ancestor? And this is how our early vertebrate ancestors developed. Fish embryos look a lot like human embryos because we mammals, we humans, descended from from other mammal ancestors, which descended from reptile ancestors, which descended from amphibian ancestors. And if you go back 500 million years, the only vertebrates on Earth were fish. So we developed the way we do because that's how our fish ancestors developed. And that's how all vertebrate embryos develop. So again, this is homology. This is embryological homology. And when we see that different species have embryos that look the same, remember, homology means there's something the same. We call this embryological homology. It's another piece of evidence that supports evolution. Before Charles Darwin proposed natural selection as his explanation for how evolutionary change occurs, he first had to grapple with the concept that species can change. Because remember, Darwin, being born in the early 1800s, Darwin accepted the thinking of his day, which was that species don't change. Every species of plant and animal we see on the earth today has always looked exactly the way it does now. Since the moment of creation, every species has existed in its present form, and species don't change. And so Darwin had to grapple with that. And he started to realize on his voyage on the HMS Beagle that all the things he was seeing didn't match up with that preconceived notion that species are static. And it occurred to him at some point in his thinking that, wow, of course species can change. There's no doubt species are capable of changing, because look at what we've done to this plant, for example. You see this picture down here on the lower left? See these little kernels? These are the seeds of the teosinte plant. Teosinte is a species of wild grass that grows in parts of South America. About 10,000 years ago, early humans started nibbling on the kernels of this grass plant, and they thought, that tastes pretty good. A little bit starchy, a little bit sweet, very nutritious. And so they started to pick the seeds from this wild grass, but they would only save the seeds from the plants that had the most kernels, the biggest and juiciest kernels, the best tasting kernels, and they would cultivate those seeds and grow a crop of teosinte. And then they would pick again only the best seeds from the healthiest plant with the most kernels, the tastiest kernels, and they did this generation after generation after generation for 10,000 years. And guess what happened? We ended up with this plant, which we all recognize, which is the modern corn plant. This is called artificial selection. Okay, this is when a human breeder manipulates the breeding of an animal or a plant in order to get particular traits that are desirable. It's where humans select which offspring to keep and to breed, and which ones to discard over many generations. And what does that do? It changes the species in the way that the human wants to change the species. Another famous example of this, of course, is dogs. We see all the modern dog breeds that we know and love. Well, we know they all descend from a common ancestor, the gray wolf. Wolves did not evolve into Great Danes and beagles on their own. They evolved in that way because humans controlled the breeding. Humans desiring different traits selectively bred those wolves to get traits that they wanted. So this is called artificial selection. And Darwin used this to back up his idea of natural selection. You know, if humans can change a species by controlling who does the breeding, then nature can do the same thing. Nature can select which traits are getting passed down and which traits are not, which of course is how natural selection works. Another line of evidence supporting evolution is what we call interspecific hybrids. That means between species hybrids. And this is where you have two different species that have chromosomes and DNA that's similar enough that they can still breed with each other. So famous example, the most famous example, I think, for the one we're most familiar with, is a horse and a donkey. Two different species. If you breed them together, you get a mule. Another example of this would be the liger, which is the hybrid that's produced when you breed a lion and a tiger. So the fact that these two species can still interbreed indicates that they must be closely related. They must have diverged from a common ancestor, fairly recently. So notice I've jumped back to an image that we looked at earlier in the lesson. I was explaining the concept of divergent evolution, where you have multiple species descended from a common ancestor. And whenever that occurs, you expect that you will find some similarities. You'll find some things that are shared in common amongst these different species. And we talked about structural homology, how their bone structures might be similar. We talked about embryological homology. All of these species should have embryos that look pretty much the same. We certainly see that with vertebrate embryos. But another thing that we would expect to see amongst these different species is that their protein structure should be very, very similar, and their DNA, the actual DNA sequence, the A's and the G's and the C's and the T's on the genetic code should be very, very similar. And if they share a very recent common ancestor, then that molecular similarity should be really, really similar. And so what I'm talking about is a different kind of homology, not structural homology, not embryological homology, but now I'm talking about molecular homology. Molecular homology refers to either DNA sequence or protein structure that is shared in common amongst different species because of the fact that they share a common ancestor. So here's a DNA study that was done some time ago, and these percentages represent comparisons between different species. So you can see that monkey DNA was compared to chimpanzee DNA, and they were looking for similarities in the actual code, the A's and the G's and the C's and the T's. And so monkeys and chimpanzees have a DNA that's 92% similar, 92% identical. And so, you know, that's not terribly surprising. They're both primates. Chimpanzees and the orangutan species, 96.3% identical DNA. Well, that's not surprising because both chimpanzees and orangutans are not only primates, but they're both what we call great apes. And apes are different from other primates because they don't have a tail. They have larger brains than monkeys, larger body size than monkeys as well. So chimpanzees and orangutans, this would be an African ape compared to an Asian species. And so, you know, it's been a little while since they shared a common ancestor, probably about 16 to 18 million years. Chimps and gorillas are both African apes, so they probably share a much more recent common ancestor, and not surprisingly, their DNA is really similar, 97.7%. But the real surprise here, maybe it's not a surprise, but I think it was probably surprising for some, is that your DNA, my DNA, human DNA, compared to chimpanzee is 98.2% identical. Well, you know, on one hand, that seems surprising that all of the differences between us and chimpanzees, you know, the ability for spoken language, the ability for technology and building and artistic expression, all the things we can do that they can't, is 1.8% of the genetic code. That seems astonishing. And don't be misled by this. This does not mean that humans evolved from chimpanzees. That's not what we think happened. But probably chimpanzees and humans split from a common ancestor, some ancestral ape species that lived, we think, about 5, maybe 6 million years ago in Africa. And so we share that recent common ancestor. Other studies have been done with protein structure rather than DNA. So in this particular study, they were looking at one of the polypeptides that makes up the hemoglobin protein. You know, that's the protein in our red blood cells that carries oxygen. And of course, fish have hemoglobin, amphibians have hemoglobin, mammals have hemoglobin, humans have hemoglobin. So you can just take that polypeptide, that chain of amino acids, and you can compare how many of the amino acids are the same in the human hemoglobin, say, compared to the lamprey. Lampreys are a group of primitive fish, by the way. And so what they found was that there's a lot of differences. Lamprey hemoglobin has 125 out of 146 amino acids that are different, you know, human compared to the lamprey. But if you compare the human hemoglobin with the frog, it's only 67 differences. Still a lot, but, you know, half as many. If you compare the human hemoglobin with the chicken, 45 differences. Mouse, 27 differences. Now we're getting into primates, the rhesus monkey, 8 amino acid differences. But if you compare this particular polypeptide with, say, humans and gorillas, which is not shown on this table, it's only 1 amino acid different. So in that 5 or 6 million years since we shared a common ancestor with gorillas, only 1 amino acid has changed in the entire hemoglobin structure. So what these numbers do is they give us kind of a clock. It allows us to pretty definitively estimate how long ago there was a shared common ancestor. So just to give you an idea, you haven't shared a common ancestor with lamprey fish in about 500 million years. That's why your hemoglobin is so different. You last shared a common ancestor with amphibians about 360 million years ago. So much more recently than you shared a common ancestor with fish. The last time you shared a common ancestor with chickens was maybe, oh boy, can't be as precise here, maybe 100 million, roughly, very roughly, 100 million years ago. And probably only about 65 million years ago since you shared a common ancestor with the mouse, and maybe 18 million years ago you shared a common ancestor with the rhesus monkey. So the more recent the common ancestor, the fewer differences we see in protein structure or DNA structure. This is called molecular homology. The greater the number of molecular differences between species indicates the greater amount of time since the last common ancestor. And if you have 2 species who have very, very similar DNA or very, very similar hemoglobin, then that indicates a very recent common ancestor. So earlier in the lesson, we talked about homologous structures. Now I want to talk about the exact opposite. So the exact opposite of homologous structures is analogous structures. And they sound just enough alike to make it very confusing for students. Remember, homologous structures, like the forelimb bones of all vertebrates, humerus, radius, ulna, etc., that's where you have the same basic anatomy, same basic bone structure that's been modified for different functions. Well, here we have the opposite. Here we have different anatomy that's been modified to perform exactly the same function. So pterodactyls used wings for flight. Birds used wings for flight, and they still do. And bats use their wings for flight. But these 3 did not evolve from a common winged ancestor. In other words, they all share wings in common, not because they split from a common ancestor that had wings. The ancestor to pterodactyls were ancient reptiles. The ancestors of birds were theropod dinosaurs. And the ancestors of bats were 4-legged rodent mammal ancestors. So from 3 different evolutionary starting points, these 3 animals independently evolved wings for flight. So this is a pattern that's called convergent evolution. When you have analogous structures that are shared by 2 or more species, not because of common ancestry, but because they adapted to similar environments, that is evidence that convergent evolution has occurred. So let's look at this concept of convergent evolution. Remember, divergent was splitting off of a common ancestor multiple species arising from one common starting point. See, this is the opposite. To converge means to come together from two different starting points. So think about ancient fish ancestors living in the ancient ocean compared to four-legged land-walking mammals. Okay, both of these would be organisms that lived long ago. But in certain parts of the earth, like the modern oceans of the earth, we have sharks and dolphins that, if you just glance at them, if you just look at them superficially, they seem like maybe they might be related. They both have fins rather than traditional limbs. They both have a streamlined hydrodynamic body shape to push through the water easily. They both have cryptic coloration, dark on top to blend in with the ocean floor, white belly to blend in with the bright sky above. All of these features make them superbly adapted for catching prey and surviving in the open ocean. But of course, sharks and dolphins, even though they share these characteristics in common, they're not related. They didn't share a recent common ancestor. Sharks are fish. They're cold-blooded. They breathe water with gills. They extract oxygen from the water. Dolphins are mammals. They're not fish at all. They have lungs. They have to come to the surface and breathe air just like you and me. They're warm-blooded. They give birth to live young. So here we have two species that have analogous traits. They have features that they share in common, not because they split from a common ancestor, but because they both adapted for life in a similar environment. Convergent evolution is the independent evolution of similar features among species that are not descended from a recent common ancestor. And so when we see analogous structures, that provides evidence that convergent evolution has occurred. So here's a few more examples of analogous structures. Butterflies have wings, birds have wings. Did they evolve from a common winged ancestor? Heck no. I already mentioned fish and dolphins. They both have fins, but they did not evolve from a common finned ancestor. And another one, I have a picture of this one, the Australian sugar glider and the North American flying squirrel. If you saw them side by side, you would think they gotta be close relatives. They must have split from a very recent common ancestor, but we know that's not so because Australian mammals and North American mammals haven't shared common ancestors in about 60 million years. The last time they could possibly have shared a common ancestor was back when all the continents were smushed together, before continental drift moved them apart. So any similarities that we see, like this little flap of skin that allows the animal to glide from tree to tree, that similarity that they both have, it's analogous. It's not homologous. It's analogous. And it results from convergent evolution. In fact, you can look at marsupial mammals, which are native to Australia and New Zealand, compared to the eutherian mammals that you find in North America and Europe. And you'll find a lot of similarities. The plantigale looks a lot like a mouse. If you saw this animal scurry through your kitchen, you would just assume, oh, we got mice, we better set a mouse trap. But this animal's not a mouse. It's not even remotely related to mice. It's a marsupial. It's much more closely related to a kangaroo than it is to a mouse. So why does it look so much like a mouse? Because it evolved to be adapted for a similar environment, a similar sort of existence. The marsupial mole and the North American mole, they look alike, they act alike, they both dig through the soil and eat grubs, but they're not closely related Any similarity between them, this is a marsupial, this is a placental mammal. They haven't shared a common ancestor in 60 million years. So any similarity has got to be by convergent evolution. This is one of my favorite examples of how confusing this can be. This animal is now extinct. It's called the Tasmanian wolf. This is a photograph of the very last one that ever existed in captivity, and I think this animal died in the 1920s. It was one of the last known Tasmanian wolves, but even before that, in the 1800s, this species was almost completely extinct in the wild. And if you saw this, if it was still alive today and you saw this run through your backyard, you would think, okay, I don't know what I just saw, some kind of mutated, looks like a cross between a zebra and a fox. But you would think, well, this is some kind of coyote, some kind of a fox, or definitely something in the dog canine family, right? It's got to be. I mean, even just looking at the shape of the leg, it looks a lot like my dog's leg. But this is not a canine. It's not remotely related to foxes and wolves and coyotes. This animal was a marsupial, like all native Australian mammals, much more closely related to a kangaroo than to a wolf or a fox. The similarities, and there are a lot of similarities between this animal and say a coyote, those are due to they're due to convergent evolution. It's not because of a recent shared common ancestor. And when we call it a Tasmanian wolf, notice we put wolf in quotes because this animal, again, did not share a recent common ancestor with real wolves. Okay, so this brings up a good question. Suppose you saw this on the AP exam, a picture of a bat's wing, picture of a bird wing, and the question is, should we consider these to be homologous or analogous? Think about that for a moment. It's kind of a trick question. I say it's a trick question because if you are referring to the internal bone structure, humerus, radius, ulna, carpals, metacarpals, and phalanges, they both have that. They have structural homology. So if I'm talking about the bones, I'd say they're homologous. The bone morphology of a bat's wing and a bird's wing, those are homologous structures. They both have the same bones because they're both tetrapod vertebrates. And as we discussed earlier, all four-limbed vertebrates have the same essential bones that they inherited from their amphibian ancestors 360 million years ago. But if we're talking not about the bones per se, but just about the fact that bats and birds both have wings, which they use for flight, that would be analogous. Because birds and bats did not evolve from a recent common winged ancestor. Bats evolved from rodents, and birds evolved from two-legged meat-eating dinosaurs. So they both ended up with wings, yes, but not because they evolved from a common winged ancestor. So the fact that they have wings is analogous. The internal bone structure of those wings is homologous. It's confusing, ain't it? Okay, let's move on. The last, well, actually it's not the last, it's the second to the last line of evidence supporting evolution is biogeography. Biogeography is the geographic distribution of species. It's looking at the patterns that we see when we examine where species are found, and does that pattern reveal anything about the evolutionary past? So here you're specifically looking at the two different broad categories of monkeys. Monkeys are kind of split into two main groups. There's the old world monkeys, which are native to Africa and Asia and Southeast Asia. And then the new world monkeys, which are found in the quote-unquote New World, right? The Americas, Central America, South America. We don't have any, at least in modern day, we don't have any monkeys living natively in North America. But the New World monkeys are distinctly different from the Old World monkeys. New World monkeys are much smaller, and they almost exclusively live in trees. They're arboreal. They live high up in the canopy of the rainforest. Old World monkeys in Asia and Africa are much larger, and there are some that live in trees, but mostly they live on the ground. Baboons, mandrills, these large African monkeys are more ground dwellers. Now, why are they so different? Well, because the last time they could possibly have shared a common ancestor, the New World monkeys and the Old World monkeys, would have been back when these two continents were all pressed together as one continent. So they've been apart, separated by the Atlantic Ocean for, I don't know, 60 million years or so. And so just that pattern, that geographic distribution of the monkey species reveals something about their evolutionary past. I'm not going to go through all these bullet points because this is another long lesson. I didn't really mean for it to be this long. I do want to mention island biogeography because one of the things that we notice when we look at island species, when we look at the plants and animals that are on especially like volcanic islands, which haven't always been there. They were formed slowly by volcanic eruptions. So like the Hawaiian Islands or the Galapagos Islands, another good example. We see species of plants and animals on those islands that are unique, that are only found like nowhere else in the world but that one island. But even though they might be unique, they always closely resemble species that are found on the nearest mainland. Now, why is that? Well, it's pretty obvious, right? Any animal or plant that lives on a new volcanic island, relatively new, had to have originated from the nearest mainland, right? And it either swam there, or it flew there, or it got blown over by a hurricane. Somehow it got out on that island from the mainland, and then it evolved. Then it changed. A famous example of this in the Hawaiian Islands is each island in the Hawaiian chain has its own unique species of fruit fly. Now, the climate is virtually the same on all of the Hawaiian Islands, so why would they have their own species, a unique species on each island? Well, probably because the oldest Hawaiian island, which has been around for 5.1 million years, probably had one species that probably got stranded there from North America in a storm or something. And then when the next Hawaiian island emerged from the ocean by volcanic eruption, some of this species got stranded on this island and then adapted to the local climate and evolved into a new species. And that trend continued even with the most recent, the newest of all the Hawaiian islands, which is the so-called Big Island of Hawaii. It has its own unique species. So anytime you're looking at patterns in the distribution of species as evidence for evolution, we call that biogeography. The last piece of evidence, and you might even say the strongest evidence that we have that evolution does occur and the process of natural selection is a real thing, is actual recorded cases where scientists have collected data and been able to show conclusively with proof that species, populations are changing from generation to generation to generation. So there's a famous example of a species of moth around the city of London, England, that within that population of moths, the predominant color has changed, has evolved from white to black over time in response to human-produced pollution, which caused the forest to become darkened with soot, and so the black moths could survive better. They could blend in with the pollution and not be eaten by predators. It's a really neat example. We talked earlier about bacteria evolving resistance to antibiotics. Over the last 50 years, 40, 50 years, there's been a gradual increase in the percent of the human population resistant to HIV, the virus which causes AIDS. And at some point, we're going to explore, and you could Google this if you're curious. There's a species of mouse in New Mexico that it has evolved over the last several thousand years in response to different colors of soil, because parts of their habitat have volcanic rock, which is very dark, and other parts have sedimentary rock, which is very light in color. And so the mice living on the dark volcanic rock have evolved darker fur color to survive, so that they're not as easily seen by predators. And there's a lot of cool studies that have been done to figure out exactly how natural selection is acting upon this population of rock pocket mice. So in closing here, I want to issue a challenge. Those of you that are seekers of extra credit, your extra credit opportunity this week is a little different. So extra credit to the first 10 to send me an email with the word Archaeopteryx in the subject line. Has to be spelled correctly. Look it up. Archaeopteryx in the subject line. And in the body of your email, tell me in a sentence or two why Archaeopteryx is considered a transitional form. That's your extra credit challenge for this week. And that is the end of our lesson. Hope you have a great day.